First speaker, we have Ben Busby from NCBI. He'll talk us about hackathons. Let's welcome the speaker.
Hey, my name's Ben. I work for NCBI. As you all know, NCBI is a fairly traditional place that houses a lot of biological data, but I do something fairly non-traditional, mostly running hackathons. I'm going to talk to you for 7 minutes about hackathons, and then at the end, I'm going to talk about why this is the title of my talk. That's essentially where we're going with the whole hackathon project. So, a lot of people call variants. We have big projects now where we have to call variants, and we got interested in being able to call variants on the fly. So, say you have a pile of fast queues somewhere, say a few 10,000 or 50,000 fast queues somewhere. Maybe you just want to call particular variants. So we built something to do that. That works pretty well. That's built on top of a new tool we have called Magic Blast, which is basically Blast for next generation sequencing. And then we're able to build robots around it. So this is basically a Dockerized module. I don't know how I feel about the strategic flipping. But basically we're able to build robots around it so you can just pass somebody a Docker container and they could call whatever variants you sent them in a bucket of fast queues they have on their local HPC. And then we can use that for looking at complex phenotypes and genotypes. And then also we can now call variants out of RNA-seq and out of single-cell RNA-seq. And so that's a pretty fun thing, and we wrap that in a Shiny app, so it's really easy to call variants out of single-cell RNA-seq. If that's something you wanted to do, if you wanted to look at concepts mentioned in PubMed about particular variants that are in dbSNP, you can do this. By the way, dbSNP just quadrupled in size. This is a really easy thing to do. Just go to fenvar.colorado.edu, and my friends remind me, they say, Ben, word clouds are not science. But underneath, you get a D3 graph of all the PubMed abstracts linked to all of those SNPs. So that's kind of a nice thing. If you're interested in new SNPs that you don't know anything about, you can look at them real quick, look at their publications. But the reason that I'm standing in front of you today, and we also built, this was with Fritz Sedlacek, we built a blacklist of variants, a public blacklist of variants for regions that have a lot of structural variants and so on. But the reason I'm standing in front of you here today is because we built all of those things in hackathons. So basically we take biological problems that are somewhat difficult to work with, and we take teams of 5 or 6 grad students and postdocs, and we build working prototype solutions to those problems. 80% of teams finish a working prototype, and about 10% of teams publish on that prototype. So that's a pretty exciting thing to me in terms of accelerating publication about tools in science. So we've done some other projects. We built a novel virus discovery pipeline. Actually, this happened in a hackathon at Pitt. That was last September. But then what we did is we realized to discover novel viruses, we should build the world's most primitive De Bruijn graph. So basically what this does is it maps reads out of SRA with Magic Blast. We do some assembly with Mega Hit or Spades or whatever. Decorate those contigs with RPST Blast N, basically with domains. And then we cut off the ends and anneal them onto the original contigs eventually, and sometimes redecorate with domains, growing the things out until we either see phages by themselves, for example, or phages in a bacterial genome. So we've wrapped that in Python, then in collaboration with Rob Edwards' lab at SDSU, started looking at it with friends. But one of the we started looking at in actual virus families. But one of the things I'm most excited about is, so we have these osf.io sites. These things have been done in a series of hackathons, and we can give everybody credit here. So not all of these projects will publish, but if any of them publish, they can give credit to everyone else, and we can get a DOI for every piece of software that we make here. So that's a really nice, I think, pragmatic way to give a lot of people credit. Obviously, lots of people have worked on this project. We built a bunch of educational resources in hackathons. This is something we built a couple of years ago with Frederick Tan at the Carnegie Institute at Hopkins. I know this is a little blurry. Besides, you shouldn't use it anyway because it's a little bit dated. But we use it in conjunction with a MOOC to teach about 3,000 people how to do basic RNA-seq mapping. So that was kind of a nice thing. Now we're really building a lot of Jupyter notebooks to do education. We build a lot of those in hackathons. Recently, we were able to build a nanopore pipeline. We think this is simple enough for high school students to use. And then recently, we also built something called Seq Academy. It's for integration of epigenomics and RNA-seq data. And we're pretty sure this is simple to use too. But what I'm really excited about is we've been able to generate really simple ways to search for viruses in metagenomes. So simple, for example, that we've taken undergraduates at SDSU and City University of New York. They've been able to dig through viruses, and actually Deloitte gave one undergrad at each institution a free semester of tuition for finding the coolest viruses in metagenomes that are sitting in public databases. So that was a really fun thing. We collaborate with a lot of people. We managed to integrate the GA4GH reads API with the NCBI reads API, so that worked out pretty well. And now we're pretty interested in building platforms. I have a poster on this, and I won't talk about it now. And also, like I said, we're interested in doing new things. So here you can take variants out of electronic medical records, run them through GWAS catalogs if they're variants of unknown significance, and then put them back into the electronic medical records. That's all Dockerized. If you're interested in getting involved, check out biohackathons.github.io. Not just ours, but the biohackathon people and some Elixir hackathons as well. Finally, what we're moving into, having modular platforms for people to build pipelines on. So we built this thing called AttackFlow. Basically, it's a next flow pipeline for AttackSeq, but it's modular so people can put in their own tools. And now we're working on frameworks where bioinformaticians can come in and build pipelines, but those pipelines are modular so people can swap in and out individual algorithms into parts of that. So that's sort of the new current push with the NCBI Hackathons project. Obviously, there's a lot of workflow systems, which makes this pretty complicated, but we can make a lot of things pretty simple by being able to pull things like Singularity and CWL into really simple to use Jupyter notebooks. If you've never seen this, this is Google Collaboratory. It's basically Google Drive for Jupyter notebooks. With that, my time is up, and check out biohackathons.github.io for more information or to get involved in the hackathon program. Thank you.
Thank you. And are there any questions to Ben?
Can you say anything about how repeatable the hackathon model is, both with Ben Busby and without Ben Busby?
As a matter of fact, thank you for asking that. So actually, there's a NCBI style hackathon in Vancouver called HackSeq. I went to the first one as a team lead, but have not been involved in the organization and running of it since then, and now they've been going for 3 years. So that's something that's quite popular. There was a keynote speaker at PAG, and all of the work she talked about started in HackSeq, in one of the hackathons. So that was really cool. Also, I've been able to hire somebody I call Ben version 2. Her real name is Alyssa Dillman, who is now running the hackathons that are on the NIH campus. So yeah, it is a doable thing. Yeah.
Any more questions? All right, with that, let's thank the speaker again.